Hi everyone, we are Group 7 and we will be introducing our experiment designed to investigate the impact of acid rain on the soil microbiome. So what is acid rain? It is any kind of precipitation that has a pH below 5.6. It can be created through natural or anthropogenic means when gases such as sulfur dioxide and nitrogen oxides react with water vapor in the atmosphere to form strong acids like sulfuric and nitric acids. Acid rain can lead to numerous problems, among which include soil acidification. It introduces acidic cations into the soil while also causing the leaching and loss of neutralizing basic ions. Soil acidification is further exacerbated by anthropogenic factors such as industrialization and agricultural activities that release large amounts of acidic pollutants into the atmosphere. The frequency and acidity of acid rain thus increases, especially in countries with rapid economic growth like India and China. Soil is a crucial non-renewable resource that provides various ecosystem functions such as food production and nutrient cycling, including carbon sequestration. The soil microbiome is especially important in sustaining majorities of soil's ecosystem services. However, soil acidification has changed the composition and reduced the function of these microbial communities in nutrient cycling, in turn leading to the loss or reduction in many essential ecosystem functions. This could result in far-reaching global impacts such as the decrease in agricultural productivity, which can threaten global food security. Soil acidification currently impacts 30 to 50% of the world's soils and has worsened over the years in some regions like Australia, where the pHCA of soil, which is measuring pH with calcium chloride, has been forecasted to drop to a minimum of 3.8 if left uncontrolled, which are considered highly acidic. As such, our study focuses on the increased occurrence of acid rain as a result of anthropogenic causes such as climate change and its impact on associated soil microbial community. Studying the impact of acid rain on soil is important as findings of such studies can be beneficial to various groups, especially communities heavily dependent on agriculture in areas that are directly affected by acid rain. Hence, the research question guiding our investigation is, how does a decrease in soil pH impact the soil microbiome in an effort to replicate the forecasted increase in soil acidification in regions as a result of climate change?
Given our research question, our overall aim is to understand the impact of soil acidification on its bacterial microbiome. To facilitate achieving this, we have a series of sub-aims. First is to cause a measurable decrease of the soil pH to simulate acidification and create selective pressure on the microbiome. Next, we hope to successfully extract and amplify the targeted bacterial DNA, as this will allow us to assess changes in the bacterial composition. Our last sub-aim is to characterize the bacterial communities in the samples pre- and post-intervention. Through these sub-aims, we can determine how acidification alters microbial diversity and composition in the soil. As we carry out this investigation, these are our expected hypotheses. Our first hypothesis is that the treated sample's alpha diversity will decrease. The addition of an acidic solution will place environmental stress on the bacteria within the treated soil, causing an overall decrease in the abundance of bacteria. As most bacteria have a very narrow range of tolerance for survivable pH, this deviation can impair cellular function via many mechanisms leading to death. A study conducted on acidified soils in North China found that alpha diversity was lower in soils with pH 4 to 5 than those with pH 5 to 6 and 6 to 7. Further, in the treated sample, it is likely that the evenness of the community will be altered, seeing an increase in the proportion of acidophilic bacteria. As acidophiles are adapted to survive in low pH conditions, addition of the acidic solution would act as a selective pressure, favoring the survival of these species and extinction of those that are not adapted. Some likely species to be observed in the treated samples are actinobacteria and acidobacteria. We also predict that beta diversity between the pre- and post-intervention samples will be increased. As the acidification will alter the types of bacteria which are able to survive, it is possible that there are species missing in the post-intervention sample that were observed in the pre-intervention sample, leading to increased beta diversity.
Therefore, our intervention will seek to simulate acid rain effects of acidification on soil by consistently adding sulfuric acid into topsoil samples over a duration of 5 days. The first part is measuring the pH in the soil samples before and after intervention. Here, 5 grams of soil sample is mixed with 20 ml of distilled water and allowed to rest before testing the solute of the mixture on pH strips. The next component is the acid itself. Sulfuric acid is chosen for the intervention as it is the main contaminant in acid rain. Global average of acid rain ranges from pH 4.0 to 4.5, and preliminary testing on the soil sample showed that it was already acidic at approximately pH 4.5. Thus, our acid solution was set to the lower bound of pH 4.0 to cause acidification of soil samples while still falling within the pH range of acid rain. This is done by gradually adding 1 m of sulfuric acid into 500 ml of distilled water and measuring the pH using pH strips until the solution reaches pH 4.0. This solution of dilute sulfuric acid is then transferred into a sterile spray bottle to simulate rainfall and allow even spread of solution when applying to soil samples. Distilled water is added into a separate spray bottle to act as the control. Next is the experimental setup. In order to simulate a natural soil environment, 2 containers were used for each soil sample, one stacked atop another to create a drainage system. The top container holds the soil sample where holes poke in the base and a thin mesh facilitates liquid drainage while preventing sample loss. Meanwhile, the bottom container collects excess liquid drained from the soil. To prevent microbial contamination, the setup was cleansed using 70% alcohol and allowed to dry before 20 grams of soil sample is measured into the top container. A lid is then loosely placed over the setup to prevent contamination but allow airflow before being placed in a location with indirect sunlight to simulate a natural environment. The final part is the adding of acid into the soil samples. Dilute sulfuric acid was gradually added to the intervention sample using a spray bottle until it was damp but not dripping wet. The recorded quantity used of 4.5 ml is then set as the dosage quantity for distilled water onto the control sample as well as future dosages. Dosage is added once every day for 5 days before giving the soil samples 2 days to dry out for the preparation of samples for 16S sequencing.
Our group was particularly concerned about the effects of soil acidification on presently degraded and acidified soil, and wanted to study the vulnerable soil in degraded forest habitats. We chose Kentridge Park as the soil there was previously used for cultivation and also found to be acidic at a pH of 5 to 5.5. To initiate our experiment, 100 grams of dry topsoil was collected. The collection was conducted using a sterile shovel into sterile Ziploc bags to ensure that the microbial communities we assessed originated solely from the soil. Topsoil was collected from underneath a tree as it contains organic material from decomposing leaf litter but also plant microbes. Furthermore, topsoil is more exposed to environmental conditions and is the layer where the highest level of biological activity takes place. Our collection methods ensure that the soil we have is ideal for observing interactions between environmental factors and microbial communities, providing meaningful insights into how acid rain affects the soil microbiomes.
Moving on to the pros and cons, one major advantage is that all of the soil samples were collected from a single site. This bulk soil sample was treated with appropriate solutions, control or acid treatment before dividing into 6 individual samples for sequencing. This helps minimize variability in the soil itself, meaning that any changes observed in the microbial compositions are associated with the acid treatment. However, this approach also introduces certain limitations. By collecting soil from only one site, which is near a tree, it may harbor a unique microbial community different from other areas of the park, potentially skewing our findings and making them less representative of the park's ecosystem. Another limitation is the removal of the soil microbiome from its natural environment during this experimental phase. Without natural factors like plant root interactions or fluctuating temperatures, microbial activity may behave differently than it would in situ. These factors are crucial for maintaining microbial functioning, and it might influence the results of our study.
There were some unavoidable limitations. The first factor we could not control was the difference between the natural conditions and the lab surroundings where we conducted our study in. This includes the light intensity, surrounding temperature, and humidity. The indoor conditions could not fully replicate the natural environment where these microbes typically reside in. Outdoor soil has fluctuating temperatures, natural light cycles, rainfall, and varying humidity levels. All of this significantly influences our treatment. Without precise control over these variables, the experiment may not accurately reflect the microbe's response to acidification under natural conditions. In order to control for the aforementioned external variables and ease of treatment, we chose to conduct the experiment indoors. Although we were unable to simulate the exact conditions of the natural park, this could have impacted the accuracy of our findings, as temperature directly affects microbial metabolic rates and their response to changes in pH. Future studies could benefit from using control environment chambers like this one from Company Lab First, where all these factors can be regulated. This reduces the potential discrepancies caused by environmental variability. Although our objective is to stimulate the effects of acid rain, it is important to recognize that natural pH fluctuations due to acid rain typically occur gradually over several years. Experimentally, these abrupt pH changes induced by our treatment could shock the microbial population and lead to microbiome extinction. While we could infer changes in bacterial diversity through the 16S sequencing, it could not cover fungi and viruses whose presence could have major impact on plants and their microbiome. Without these measurements, our understanding of the shift in the microbial community will be limited. Other sequencing methods such as internal transcript spacer, ITS, can be implemented to get more information. These measures would give us more representative results and accurate conclusions.
In conclusion, our experiment involves treating topsoil samples with diluted sulfuric acid to investigate the effects of acid rain on the soil microbial community through 16S sequencing. Thank you.